So my lab is really interested in understanding how the brain encodes, stores, and uses information. We think this is, of course, a really fundamental process of the brain. And we think that this is one of the fundamental processes that can go wrong in a variety of different diseases. And so I think that it's really cool to study because it's like this important process, but because it also may have down the road important clinical implications. And I always like to start off with either a Drake quote or something from, Aristotle and Plato. So the idea that memory is stored as enduring changes, possibly in the brain, dates back long, long, long ago, before even I was born. But I'm going to be talking a lot today about ideas that were pioneered by Richard Semon, who was a memory theorist long ago. He published his first book in 1904, and he is the one that's credited with coming up with the term engram. So like it or don't like it, I'm going to use it today, and we can quibble about if it's a good term to use, if it's not a good term to use. And I'm going to use the definition that he put forward because I love it. It's the enduring, though primary latent modification in the irritable substance, the brain, produced by a stimulus. And I love that definition because it sort of says everything, but doesn't really tie you down. And he also came up with this other amazing definition of this word that he made up called ekphory, which is basically memory retrieval. And he defined it as awakening the engram out of its latent state into one of manifested activity. So after Richard Semon came up with these terms, you probably all know, if you've taken Psych 101, that Carl Lashley spent about 30 years of his scientific career searching for an engram in the brain, and he couldn't find an engram. And he basically said that the engram is elusive. But when I started my lab, I wanted to sort of continue on in this sort of trajectory and say, well, let's mix things up just a little bit. I thought that Lashley was onto something. I thought that Semon was onto something. Why don't we change it a little bit? So for my search for the engram, I'm going to choose a different form of memory. So I'm going to use a very, very simple form of auditory fear conditioning. And in this paradigm, super simple, one-time learning, we're going to pair a simple tone with a foot shock. It's not, of course, enough to harm the animal, just enough for an animal to say, what the heck just happened here? Then at any point thereafter in the lifetime of a mouse, so it's up to 2 years, we can place a mouse in a new context, replay the tone, and if that mouse remembers the last time they've heard that tone, it produced this aversive foot shock, they're going to show us that they remember by freezing to the tone. That's a species-specific defensive response, and it's super well characterized, which is why we're going to use it. So our first question was, well, if we think that our engrams in the brain that represent events or experiences, what is the architecture? What does it look like in a given brain region? And we sort of said, okay, there's a sliding scale. It could be a dense code where every neuron's involved, all the way down to the sort of one neuron in a given brain structure, sort of like your grandmother's cell, is involved in representing this experience. And of course, you've already heard that it's probably sparsely encoded. A lot of work by many, many pioneers have suggested this. So we said, okay, if our engram is going to be sparsely encoded, how are these neurons recruited or allocated to the sparse engram? Why is this neuron part of this engram and this neighboring neuron not part of the engram? So we did a bunch of experiments, and I'm going to just summarize them here. So what we did is we looked at in the lateral amygdala, which is a key area for this auditory fear conditioning memory. And what we did is we used viral vectors. We started off with herpes simplex, but any viral vector will do as long as it infects a sparse, random population of excitatory principal neurons in the lateral amygdala. So we have these our viral vectors, and they're going to randomly infect a sparse population of neurons. And what's going to change is that they're all going to, throughout different experiments, the payload inside this viral vector is going to change. So we're going to overexpress CREB, we're going to express different potassium channels, we're going to express optogenetic or chemogenetic constructs. And the one sort of unifying thing between all these disparate constructs is that they all serve to increase the excitability of these neurons that we've infected. Then we are going to train the mouse in auditory fear conditioning. And then right after we test the mouse, we're going to look in the brain to find the neurons that are active. And we started, this initial work with John Gazowski, where we did ARC catfish to look at neurons that have nuclear localized of the mRNA for the activity-dependent gene ARC. And neurons that are active, we're going to suppose are really important in storing and retrieving this memory. And for better or for worse, we're going to call these neurons that are active engram neurons. And what we found when we did these experiments, despite all these different cargoes, and they work in all sorts of different ways, we found that we were able to bias these infected cells, not 100%, but we were able to bias their inclusion into the engram supporting this auditory fear memory. And then we did exactly the reverse experiments. Same setup. Oh, I forgot to say that although we did a bunch of different things, we found that the size of the engram never seemed to change. So the number of these engram neurons seemed to be constant. And we observed it, we really love fear conditioning because it's quick and easy, but we also observed it for an appetitive memory. So we also did the exact opposite experiment. So now we're going to have our viral vectors, and the payloads are either going to be a mutant form of CREB or a potassium channel, inwardly rectifying potassium channel, which serves to decrease the excitability of these cells. And what we found then was, well, what happens when we decrease the excitability of a similar small population of random cells in the lateral amygdala? Well, we hypothesized that nothing at all would happen to memory because the remaining cells that presumably have normal levels of excitability would be sufficient to be part of it, to form an engram for our fear memory. And this is exactly what we saw. But interesting, what we really saw was that we were able to bias the exclusion of these infected neurons away from the engram. So again, when we looked at the memory for these mice, the memory was normal. Again, the size of the engram did not change. So we were able to bias whether a neuron went into or was excluded from an engram just by artificially manipulating the excitability of these neurons. So then Asim Rashid, who is a research associate in my lab, said, well, are these neurons that we're allocating, are they really necessary for memory retrieval? So he performed a very cool experiment, which took advantage of a construct that we got from Carl Deisseroth. And this is a really lovely picture of Carl Deisseroth and myself. And look at this, 2016, Carl Deisseroth, such a visionary, such a leader, social distancing from me way back in 2016. And now everybody's socially distancing. So just follow what he does. So what this construct is both, it expresses both channel rhodopsin and halorhodopsin. So that allows us to either increase or decrease the activity of the same infected population of neurons by simply shining different wavelengths of light. So when we shine a blue light, we're going to activate channel rhodopsin, we're going to increase the excitability of our infected neurons. Then later on in the experiment, we can shine red light, activate halorhodopsin, we're going to be able to silence this exact same population of infected neurons. So here's what Asim did. So he's going to take our viral vector that expresses both channel rhodopsin and halorhodopsin. He's going to microinject it into the lateral amygdala. He's going to infect a sparse population of random neurons. And right before we train the mice, he's going to turn on the blue light, excite these infected neurons, and try and allocate them into the resulting engram. He's going to train the mice, and he's going to test them. So how do we know that these neurons are necessary for this fear memory retrieval? Well, he's going to test them under two conditions. He's going to test them both when we turn off these infected neurons and also when we don't. Just see what normal memory looks like and what memory looks like when we decrease the activity of these infected neurons. And when he does this, he sees that we see really good levels of memory. Here's our percent freezing to the CS, really good levels of memory in the normal condition. When he turns off these cells, he sees a disruption of memory, so they don't freeze as much. Well, it could just be turning off a small population of cells in the amygdala disrupts memory. So he did a control experiment where he did the exact same thing, but he didn't allocate the infected neurons into the engram. He didn't shine a light before training, but he turned them off during the memory test. And when he does that, he sees no effect at all. So just disrupting the activity of a small population of random cells in the lateral amygdala doesn't do anything to memory. So then along came Sung-Mo Park, and he said, well, this is really cool. He's a postdoc in my lab. He said, oh, so these neurons are necessary for fear memory. Are they also sufficient? So this is the experiment that Sungmo did. He used either HSV expressing channel rhodopsin, or you can use AAV with CAMCase2 promoter. Again, he's going to infect a small population of neurons as before. He's going to shine the blue light, trying to increase the excitability, trying to bias these cells into the engram supporting this memory. We're going to train the mice, and we're going to test the mice. Now, typically, when we test the mice, we give the mice the perfect retrieval cue, which is the tone. So the tone that predicted the foot shock. And we think that this tone goes on and reactivates these engram cells that we allocated. So what Sungmo is going to do is trying to take out the middle person in this. So he's going to not give the mice the tone, the perfect retrieval cue. Instead, he's going to go in and directly reactivate these neurons he's tried to allocate into the engram. So during the test session, he just places mice in a novel context. He sees what their freezing is like normally, and then he's going to shine the blue light to try and reactivate these engram cells, see what happens to memory, then he's going to turn it off. So we think this sort of goes back to what Semen's original definition of ekphery is: awaken the engram out of its latent state into one of manifested activity. But he's going to do it optogenetically. So this is what the data comes out like. When you just place mice in a novel context, you get very low levels of freezing. Sungmo turns on the blue light. We see freezing behavior. He turns it off. It goes away a little bit. He turns it on. It comes back a little bit. So we think this might be a way of artificially activating the engram to cause sort of artificial ekphery. Now, there could be differences and similarities between the levels of freezing and the types of freezing, between the sort of natural retrieval and between artificial retrieval, and we're sort of looking at that. But this suggests to us that these allocated cells are really important in retrieval of the memory. So this left us with the following hypothesis. So we thought, well, okay, eligible individual lateral amygdala principal neurons seem to compete against one another for allocation to an engram supporting an auditory fear memory. So we suppose that all these neurons start out equal, but this one neuron wins this competition against its neighbors because it seems to be more excitable at the time of the training event. So then we said, okay, this is really cool. We can do all these optogenetic tricks, these chemogenetic tricks, but the real question of the day is, does this excitability-based allocation process occur endogenously? And if it does, is the individual neuron the basic unit of allocation? So we looked in the literature with our good friend and colleague, Morris Moscovich, and he published this really lovely paper which looked at something that could be sort of akin to allocation in humans using fMRI. And he found some sort of data that was consistent with this, but he looked in the human equivalent of the dorsal CA1. So we're going to change from looking in the amygdala, and now we're going to look in dorsal CA1. So, of the hippocampus. So our first question is does this excitability-based competitive allocation process happen in the CA1? And these are studies that were done by Adam Ron-Saran, who is a graduate student in my husband Paul Franklin's lab, and this is how I usually see him on our many Zoom calls. He looks very sleepy. So what he's going to do is trying to do this artificial allocation, much in the way that Assam did in the lateral amygdala. So again, we're now going to change the context fear conditioning and look at dorsal CA1. He's going to infuse 2 groups of mice with our construct that has both channel rhodopsin and halo rhodopsin, infect a small population of neurons, train the mice, and test the mice. And he tests the mice under 2 conditions. So without the red light, without turning off these cells, and when he turns off these cells that he's allocated, to see if they're really important in the engram supporting this memory. We would hope to see a memory reduction, and this is exactly what he sees. So in our non-allocated group, turning off a small bunch of cells in the dorsal hippocampus doesn't do anything to context fear memory. But in our allocated group, so we've increased their excitability, we're trying to allocate them into the engram supporting this memory, we see a disruption when we turn off their activity. So this again suggests that this similar excitability-based allocation process can be artificially induced, at least in dorsal hippocampus, the CA1 region, during the formation of an engram supporting a context fear memory. So what we want to do is say, okay, let's identify endogenously allocated neurons during the test and really simply just see if they're more active or more excitable than their neighbors before the training session. But we don't really know what the signature of an endogenously allocated neuron is during our memory test. So we're going to start off by looking at examining the signature of an artificially allocated neuron during our memory test. And for this, we're going to use bulk fiber photometry signals. So we're going to be able to look at bulk calcium from GCAMP expressing cells from neurons that we artificially allocate to an engram. And for this, we're going to use, so our GCAMP signal is going to take up the blue and the green channels. So what we need is a red light sensitive optogenetic exciter. And these studies were done by Andrew Mokley and again, a sleepy looking Adam Ramsarn. So what we're going to use is another construct from the Diceroff lab, this that has a red light sensitive optogenetic activator and GCAMP. So again, really similar to before, we're going to express this construct in a small population of CA1 neurons. We're going to have two groups. One, we're going to allocate these infected neurons into what we think is the engram supporting this contextual fear memory, and another group, just random neurons. But then we're going to just look at their GCAMP activity during the memory test. Are they different? Importantly, in these two groups, we see very similar levels of memory. But what really was different between these two groups is their level of GCAMP signal right before each freezing bout. So each of these dotted lines is a freezing bout in the test session. And with this mouse, you can see this ramping activity of GCAMP signal right before each memory, each freezing bout that we do not see in the unallocated group right there. So when we set everything to all the freezing time, we put all the freezing times at time 0 and we do a normalized fluorescence, we see all of the allocated neurons show us this ramping activity right before the freezing episode, and we don't see this in the unallocated group. So when we sort of parse this out, we see this large difference in GCAMP fluorescence in our allocated group, but not in our unallocated group, in this roughly 2 seconds right before a freezing bout, and we see no difference at all during the freezing bout. So we took from this that the neural signature of an engram neuron during our memory test is going to be this increase in calcium signal, which is our proxy of neuronal activity, in about 2 seconds before each freezing bout. And we started looking at this and we said, 2 seconds before the freezing bout, that's not exactly the time scale we were thinking of. But then we looked in the literature and we were reminded of this very cool paper that came from Rafi Malik's lab at the Weizmann Institute. And here he's looking at human patients with epilepsy, and they have electrodes implanted in their brains trying to localize the source of the seizure. And he did a memory test with them, and what he found was similar to what we found. So approximately 2 seconds before free recall of a memory, you saw this increase in the sharp wave ripple rate. And you could sort of say a proxy of sharp wave ripple rate activity might be the discharge of about 10 to 15% of pyramidal neurons in the CA1. So our timing was really, really similar, even though we were looking at very, very different species and tasks. So that gave us some confidence that we were actually looking at something that was real. So this is, bulk calcium signature from a bunch of neurons. What we really want to see is this at the individual neuron level. So we're gonna, Denise has told you about her UCLA miniscope adventures. I also came from Alcino's lab way back, and this is Chen Yan. He's used to be a graduate student in my lab, and he made his own miniscope. His name is Chen, so we call it the Chendoscope, and we published a bunch of papers about it. So these are final experiments, and I'm running out of time. I'm gonna get the chicken, where we looked at this. I'm burying the lead. So we have miniscopes in these mice, and we're gonna do exactly the same thing. We're gonna apply our definition of engram that we got from our fiber photometry studies. We're gonna identify these engram neurons and just ask, are they more excitable right before the training period and also 24 hours beforehand? This is, again, our definition of an engram neuron. This is one example of a neuron that we defined as an engram neuron. We have engram neurons and non-engram neurons that we're going to identify during our test session. They look just like our definition, which isn't all that implausible. And what we see is when we look back, we have engram neurons and non-engram neurons that we identified during the test. Whoops, almost finished. And we have also another population of neurons that were undetected in the test, but we saw in the training period. We're going to use OASIS spike deconvolution to look at their activity in the training session. And what we see is they are more active. Neurons that went on to be identified as engram neurons during the test were more active right before the training session in this 5-minute period, but not in the home cage. So we think this is evidence for endogenous excitability, which mediates, again, this endogenous memory. So with that, I just want to thank all of my collaborators and my funding agencies, and I'm sorry for going over time. Thank you.